脑人言。
大家上午好，国内的朋友，上午好，然后呢，美国的话，大家晚上好，欢迎来到这个 Neureport 第75期。今天呢，我们非常荣幸邀请到王乐博士。王博士呢是中南大学跟 Rutgers University，也就是罗格斯大学的联合培养博士，现在呢是 UCSD 加州大学圣地亚哥分校的博士后。他的主要研究方向是进食行为的神经环路以及神经态突触调控机制的研究。最近呢，GLP-1药物非常，今天呢，王博士呢给我们带来一个报告，叫做中枢 GLP-1 神经突触调控机制。OK，the floor is yours。
对，
Go ahead。
好，OK，我 check 一下我的 slide。OK，感谢郑刚，当然也感谢脑人研也提供这样一个机会来介绍我们最近发表在Nature Metabolism 上的工作。当然这个整个 presentation 我可能用英文来 present， 但是接下来的那个提问的环节，你可以用英文或者中文来提问，我尽量就是用中文来回答。OK，let's get started. Thanks, Zhenggang, and I appreciate I have this opportunity to introduce my recent work. This work actually recently just published on Nature Metabolism. First of all, I want to briefly introduce obesity. So the first slide showing here actually is the data. Recently, I just got it from WHO. You can see the global population level data from through UK as well as US, and more importantly, I want to emphasize the data actually from China. Is at the population level we actually observed in last about like 30 years. There's a significant increase in population level. People actually generally getting more overweight and obese. So obesity right now actually become a serious, actually very important global epidemic phenomenon. So for treating obesity, the people you may don't know what is GLP-1, and I believe some of the audience you must know what is Ozempic or semaglutide. So Ozempic or semaglutide right now really become a rising star, a famous drug in the pharmaceutical industry as well as in the clinical trial. So because it's robust, the body weight and reduced blood sugars effect actually has a large impact in the field. So Ozempic or semaglutide actually represent a group of drug actually targeting endogenous peptide, so-called GLP-1, glucagon-like peptide 1 receptors. So endogenously, GLP-1 as a peptide actually encoding in our genome is encoded by the GCG genes. And by the early work, actually, physiological function of GLP-1, we already know is can lower the blood glucose and suppress the appetite or suppress the food intake. That's a major reason actually GLP-1 right now serves as an important drug target for both diabetes and obesities. So you can see in the right side of this panel, showing here actually nicely summarize the development of the GLP-1 and the GLP-1 analog in the past more than 20 years. You see through early 1980s, after first identified what is the GLP-1 actually from lizards. So, and the following year, they actually synthesized the GLP-1, first generation of GLP-1 agonist, examining 4. And in the past 20 years, you can see actually frequently there's a lot of effort actually in pharmaceutical industry, as well as fundamental basic research, trying to push the boundary of develop a new generation of a GLP-1 receptor agonist. So, because the impact of the GLP-1 agonist in body weight control and blood glucose control in 2023, I really want to emphasize here. So because the work of the GLP-1 analog, so in the science journal, actually GLP-1 analog work being acknowledged as a year of a breakthrough study in 2023. And more importantly, you can see in the right side panel showing here, so last year, these are 3 pioneer scientists in the field actually, because they are pioneer work and their effort in identify the GLP-1, as well as the effort to push the boundary of developing new agonist analog of GLP-1 receptors. So their work actually being recognized and acknowledged as a 2024 Lasker Award. And meantime, I want to emphasize those 3 pioneer scientists. They are not only from academia, and they also from the pharmaceutical industry. So a lot of clinical data are actually very optimistic, and there's a strong effect, a lot of money entering in the market, so entering to develop a new generation GLP-1 analog. So the question really become can we end up the obesity epidemic by using the GLP-1 analog? So the answer I would say right now is no, because actually through this news in the social media, actually also as well as in the clinical trial in development and the clinical trial of GLP-1 analog, we already know the current GLP-1 analog actually associated with many side effects, including one of them so-called Ozempic. So these side effects actually including, I would say, in portion, not all of them, but in portion of a patient, they actually have a severe aversive effect, for example, like a nausea. And there's also some literature reported that GLP-1 analog in clinical trial also induce some mental issues. For example, the patient actually form a higher ratio in having depression, and as well as there's some reports saying GLP-1 analog can induce a suicide behavior. So this actually these are actually those are like a side effect really make the space actually to create, we want to create a new generation, better GLP-1 analog to improve the, to benefit and the effect of analog in using treat obesity and diabetes. So to addressing these questions or create a new generation GLP-1 analog, I think the most important thing is to understand the fundamental function of endogenous GLP-1 peptide, and also as well as the endogenous GLP-1 analog, how it's working in our bodies. So through these slides, I wanna emphasize here is originally actually GLP-1 be identified is a gut hormone. Actually, many being released in the gut L-type cell. Although there's some like a literature report, there's a gut-brain body-brain interactions. The gut L-type cell actually release GLP-1 interact with both peripheral nerve system and also central nervous system. But in our brain, in the central nervous system, there's a unique, very unique small population neuron located actually in our hindbrain, the region so-called NTS. There's small population neuron actually can release neuron peptide service as a neurotransmitter, moderator our behaviors. So we're talking about the brain. So actually, the diagram showing, the images showing here, the human brain actually is a very complex organ. I think everybody in the audience know. So the human brain actually over 1,000 grams. So according this classic work by Roman Carhar's characterization, there are more than billions of neurons in our nervous system, and they have actually formed the trillions level of synapse from the dysfunction units in our nervous system, control our emotion and behaviors. So the diagram showing here actually is a very simple diagram, showed the basic unit, what's the structure of the synapse. So it contains, simply say, it contains a pre-synaptic component and a post-synaptic components. And the basic unit and the basic function of a synapse and the synaptic transmission actually is extremely important in control, for example, our learning memory, decision-making, movement control, as well as our eating behavior. So however, when the synaptic transmission dysfunction, so those dysregulation actually highly associated with many disorders, including many mental disorder like autism or schizophrenia, And as well as there's also many evidence showed the synaptic dysregulation also associated with obesity and many eating disorders. So I think to understand the detailed molecular mechanism of how does a synapse function, how does presynaptic the vesicle being released, there's in the field, there's already have a lot of literatures. You can see this, I think, is a mostly simplified version how to describe what happened in the synaptic terminal. So it's actually contain presynaptic terminal, those neurons have to generate electrical signals to generate the action potential. Following the action potential, there's a calcium influx in the presynaptic terminal, and further to trigger the synaptic vesicle release. Those vesicles actually contain those chemical signals, and the chemical signal can further release to the postsynaptic component and binding interact with their specific receptors. Some of them, especially those typical fast neurotransmitter, can generate this postsynaptic currents. So as I just briefly described, this process is relatively complex, but when you look at the time scale of all this process actually only happened in the millisecond level, that's actually indicated to me is in this short time window, the position or precise control of synaptic transmission actually is extremely crucial. So through the elegant work, there's a lot of pioneer, including my mentors, and he was working with Thomas Sudolph and spent a decade of time studying the molecular mechanism of synaptic vesicle release. And through their literature review summarized showing here, actually it's very brief version. There's a family of a protein complex, actually so-called snail complex. So there's a snail complex in the nerve terminal actually functioning in recruiting synaptic vesicle in the nerve terminal and help those synaptic vesicle docking in the active zone and help them release the neurotransmitter neurochemical signals. And also they are functionally important in recycling also synaptic vesicle in the terminal. So those are detailed molecular mechanisms already examined in the synaptic vesicle release. However, when you look at the left side diagram showing here, so in the nerve terminal, there's not only small synaptic vesicle, there are also the other group of vesicle also exist in the nerve terminal. For example, like a LDCV, so-called a large dense core vesicle, also exists in the co-localized or co-exist in this terminal. And I'm showing here actually is a beautiful EM structures actually I adopted from a pioneer neuroscientist, Van der Poel. He was Yale. So his elegant work actually examining how the densical vesicle function in his whole scientific journeys. But you can look at these beautiful EM structures and point out here this SV. So synaptic vesicle actually highly enriched high density in the nerve synapse terminal. But there's also, I want to point out that there's in similar regions, there are also the other group of densical vesicles actually compared with the synaptic vesicle, they contain actually different neurochemical signals. So typically, those synaptic vesicle contain is a faster neurotransmitter, including glutamate or GABA. They can generate the postsynaptic currents. But however, those densical vesicles, normally they contain a lot of different type of neuron peptide, actually including the GLP-1, what my current study or focus on. So we are talking about the neuron peptide. So those neuron peptide actually has a distinct function and a distinct kinetic in our central nervous system. You can see that this diagram showing here compared with a typical fast neurotransmission like excitatory, glutamate release, or GABAergic inhibitory release. So those neuromodulator neuron peptide actually functioning kinetically is much slower. And their function actually mainly through a group of receptors so-called GPCR expressed on the memory. So they are functioning not directly deeper the cell, but they're functioning actually moderate those faster neurotransmission release, for example, glutamate release or inhibition tone in our nervous system. So through those background information, actually really narrow down our research interest. We actually trying to understand the neuron peptide, the GLP-1. How does the GLP-1 in our nervous system, actually those GLP-1 contained by the large densicle release, how does the GLP-1 being released in our nervous system? And how does the GLP-1 functionally as a neurotransmitter or neuromodulator modulate the faster neuron transmissions in our brain? Actually, it is extremely fascinating question for us. So I was introduced the GLP-1. Actually, I want to emphasize here again, because the GLP-1 endogenously is encoded by the GCG gene. So we're trying to understand the endogenous function of the GLP-1. So to get the genetic access, we actually get the genetically modified animal, so-called GCG Cree. So with this genetically modified animal, we can genetically specifically manipulate those GLP-1 synthesis or GLP-1 releasing neurons. So in this case, by injecting Cree on channelopsin virus in the hindbrain, as I introduced before, this spatial localization of those GLP-1 releasing neurons, you can see through channelopsin UIP virus, you can label this hindbrain releasing neurons. And through systematic, actually morphological mapping, we actually figure out in the forebrain, there's one particular region stand out, so-called PVH or PVN, paraventricular hypothalamus. So this region actually receive, you can see the imaging showing here, receive a high density of axon input from hindbrain. In the meantime, we also use another Cree, so-called GLP-1 receptor Aris Cree. And this Cree, I use a lot in my study. So I want to introduce here. But here, we actually use the GLP-1 Aris Cree crossed with the report line, so-called AI14. So with this genetic cross approach, we can genetically use TD tomato to label the GLP-1 receptor neuron across the whole brain. But in the PVH, I want to emphasize here, so in the PVH, actually, you can look at the images. There's a highly abundance of GLP-1 also exists in receptor neurons, also exist in this forebrain or hypothalamus region. So at least the information I want trying to say here is through the morphological virus tracing and the reporter line characterization, we want to say morphologically, this PVH potentially will be very important hotspot to study endogenous GLP-1 signaling in our brain. So meantime, I also want to show emphasize the work actually conducted by my previous lab member, Ji Liu. Right now, he is a professor actually in UTSC. So his work actually 2017 published on Neuron actually beautifully resolved this hindbrain, I would say, ascending circle, those hindbrain GCG or GLP-1 releasing neurons and the robust protection to the forebrain PVH region. And his study really resolved a post-synaptic current mechanism. So the GLP-1 be releasing in the PVH and can enhance the post-synaptic currents, excitatory currents in control animal food intake behavior, also maintain our body weight. So we actually, through the systematic study and my research actually really just follow up what G have done before. So I'm trying to answer a very simple question at the beginning. So what's the downstream target of the PVH GLP-1 receptor neurons, and what are the functions? So to answer this question, again, we are able to use this GLP-1 Aris Cree line as access to get access to these GLP-1 receptor neurons. In very beginning, the first experiment, I inject again Cree on channelopsin UFP virus in the PVH. You can see label in the middle panel showing here. So I did whole brain mapping experiments, and there's one brain region actually again stand out. This region actually received the most abundance GLP-1 receptor neuron in PVH, their projections. So this nucleus is so-called, actually it's a nuclear complex, so-called DVC, dorsal vagal complex. So this region actually contains 3 different nucleus, is including motor nuclear, dorsal motor vagus nucleus, as well as the NTS. Actually, it's very interesting. NTS is especially this caudal NTS actually exactly this GLP-1 releasing neuron localized here. And another region also very important actually in the I would say pharmaceutical side is associated with pharmaceutical side effect of GLP-1 analog, so-called AP aversive tremor. So this brain nucleus actually functionally associated with this aversive nausea effect. So our lab actually expert in synaptic physiology or electrophysiology. So through combine this virus tracing approach and optogenetic technique and the slice physiology, we're able to get access. We inject the channelopsin in PVH, and we're able to record those post-synaptic neuron in the DVC. So by shining the blue light, activated this presynaptic terminal can trigger those nerve terminal to release neuron chemicals. So in this case, I can robustly record those post-synaptic current following these optogenetic simulations showing in middle panels here. And this response actually robustly 100% can be blocked by the antagonist CNQX. So this CNQX actually is AMPA receptor antagonist. That's actually information tell us is this descending circle actually functioning as an excitatory descending circle. Those GLP-1 receptor neuron, not surprisingly in PVH, they can release glutamates. And through another experiment, actually we did is just sort of random patching around the DVC across DMV, NTS, and aerospace tremor regions. So by quantifying the sort of successful rates, how many cells are responding to the lights, and their absolute value of this lights triggered post-synaptic currents, we are able to conclude about this piece of data in the DVC region. So this motor nucleus, so-called DMV, actually predominantly received this excitatory input from PVH GLP-1 receptor neurons. So to further verify these tracing experiments, we also conduct many experiments. This one of it. So we also conduct retrograde tracing again by using GLP-1 receptor RSCRE. We inject a retrograde on AAV UIP reporter virus in the hindbrain DVC regions. So by systematic quantification in this retrograde tracing, we're actually able to conclude, you can see in this represented imaging showing here, as well as this quantification. So this descending circle actually is manning, this descending circle actually is corresponding to the caudal part of PVH. They form these descending glutamatergic projections. So next, the question that we really first trying to understand what's the function of this descending circle. To address the function of this descending GLP-1 receptor neuron in PVH, we actually designed a projection-specific or pathway-specific strategy to selectively manipulate these descending GLP-1 receptor neurons. You can see this diagram showing here. So again, we are using GLP-1 receptor RSCRE in this case, and then we are injecting CRE on retrograde Philippi virus in the hindbrain DVC region. So the CRE will turn on this contrast, this Philippi expression will retrograde back to the PVH regions. So in the PVH, in this case, we are injecting the Philippi-dependent HM3DQ DRAS virus. So by using this approach, we can selectively chemogenetically activate this descending circuit, GLP-1 receptor neuron, that are only from projection to the DVC regions. So first of all, the control experiments we confirmed by adding, by slice physiology, we call those like retrograde chemogenetic HM3DQ selectively labeled neuron in PVH by applying their DRAS ligand, so-called CNO. So we are able to observe that with this approach, CNO actually can significantly depolarize the cell memory potential, depolarize these cells, and we also observed that those neurons increase their firing frequency. So we actually use this approach, conduct a bunch of behavior screening But really, it comes out the most dramatic thing is when in the freely moving animal, when we IP inject the CNO to those animals. Actually, I'm showing here just a piece of data, but actually in different motivation states or energy state, whenever the animal getting fed or fasted overnight, the animal actually statistically significantly suppress their overall food intake levels. So by alternative approach, also further verify this data, we also conduct a optogenetic approach to verify this data. By using optogenetic, in this case, we are using injecting the Crea-on channel dopsin virus in the PVH. And meantime, we also implant the optic fiber in the hindbrain regions. With this approach, we can selectively activate the nerve terminal. Actually, those GLP-1 receptor neuron project to the DVC regions. So very similarly, first experiment we conduct is a freely moving assay. So just photo simulation, this descending nerve terminal compare with our EVA FP control group when we shine in the light actually with 20 Hertz simulations. So in the channel dopsin group, we observed there's again significant surprise in food intake level in channel dopsin group. So we're able to conclude actually selectively manipulate, activate this so-called GLP-1 receptor neuron. Actually, they're corresponding to association, I would say association signal largely suppress the food intake level. So in the meantime, because this neuron express the GLP-1 receptors, and we want to understand how does the GLP-1 signaling and function in these GLP-1 descending projection neurons. So to understand these questions, again, we use what we expert on the synaptic physiology by injecting channel dopsin again in PVH. We cut the slice and we recording the DVC post-synaptic neurons. But in these experiments, we actually recorded those post-synaptic neurons by shining the lights, recorded the baseline. And in the meantime, after the baseline recording, we applied the drug so-called Exanin-4. As I mentioned previously in the previous slides, Exanin-4 also is an agonist for GLP-1 receptor neurons. In the first set of data, we actually observed when we applied the Exanin-4 in the bus, we observed that this Exanin-4 actually increased the AMPA receptor median EPSC amplitude. And to further understand what happening in this nerve terminal, we actually collected other paradigms. So here, I'm showing here is we also meantime recorded the power pulse ratio paradigm, and we also recorded the AMPA receptor, MDA receptor ratio paradigms. So by this piece of data, we observed this power pulse ratio we observed after Exanin-4 perfusion. So there's a significant decrease in power pulse ratios, and there's no change in AMPA and MDA ratios. So these two pieces of data really tell us this activation of Exanin-4 activation of GLP-1 receptor neurons really seems through a presynaptic effect. Seems through when we selectively activate those GLP-1 receptor neurons from PVH to DVC, it can enhance the synaptic vesicle release probabilities. In the meantime, is in similar data experimental design. We know this GLP-1 signaling through GLP-1 receptor is a GPCR protein functioning through. It's being reported functioning through CAMP and the PKA pathways. So a blocking experiment actually we conduct actually here is we wondering whether PKA signaling is also involving in this enhancement of synaptic release. So we actually pre-incubated those slides with like an hour incubation with a drug so-called H89. It's a drug actually is a blocker for PKA pathways. So after this bus incubation of these slides, we conduct very similar experiments again. So basically we recorded the baseline and we applied Exanin-4. But when we selectively block the PKA pathway, we actually the activation or enhancement of a glutamate release, the phenomenon is gone. So thus, this piece of data here showing here tell us is this activation or enhancement of glutamate release actually through CAMP PKA pathways. So meantime, also to further confirm these experiments, we also conduct elegant strong-syn experiments. So in this case, we are use a 5 millimole strong-syn buffer to replace 2.5 millimole calcium buffer, because we know the synaptic vesicle release actually require calcium activities. But in this case, when we apply the strong-syn, actually we are delayed you can see the first peak, we call a synchronized synaptic release. So when the strong-syn applied and replaced with a calcium, you can decrease the synchronized release and increase this sort of slow, unsynchronized slow response. By this approach, we can really quantify the quantum release of a pre-synaptic pool. So very similarly, on slice of physiology experiments, we recorded this response from PVH to DVC, and again, with a 5 millimole strong-syn, we applied the exanin-4 agonist. So with this approach, we actually quantified both a synchronized response and a synchronized response, slow response, their frequency. So we're able to observe that there's no amplitude change in synchronized response, but we actually observed a very strong increase in frequency of this slow response. Again, put all these synaptic physiology data together, we're able to conclude actually in this descending circuit from PVH to DVC, the presynaptic terminal GLP-1 receptor actually regulates synaptic transmission through increase the presynaptic terminal quantum release or or synaptic vesicle release probability to further enhance the glutamate release responding to the postsynaptic in the hindbrain postsynaptic neurons. So the next questions we're wondering, how about in vivo? How does this descending neuron, GLP-1 receptor neuron, they are natural dynamic in responding in different behavior. So to address that, we actually similarly we use this similar design of a projection-specific strategy by using GLP-1 iris-Cre and injecting retro flippy virus, Cre-on flippy virus in hindbrain, and in the hypothalamus PVH. In this case, we are expressing the GCAMP indicator, so-called GCAMP6 in the flipping on GCAMP6 in the hypothalamus PVH regions. In the meantime, we also implanted this optical fiber in PVH. So by using this strategy, we can selectively recording those calcium, those neuron transient calcium response in vivo. So we did a actually very simple behavior paradigm I'm showing here. Actually, we just manipulated those animals by simply just overnight fasted those animals compared with well-fed condition of those animals. We actually directly present a food as a food cue, directly present those food to those fasted animal or well-fed animal. In control experiment, we actually present is a very similar shape of a control object, very similar shape as a food cue directly in these two conditions of freely moving animals. But you can see in first the quantification of the heat map showing here, also the bar graph showing here. So in vivo, this neuron calcium transient actually selectively only responds to the food cue in fasted conditions. They are not increase their calcium response when those animals being well fed, and also they are not increase their calcium activity when the animal fasted, but actually received this control objective cue. So in the meantime, we also actually followed by the other group. There are studies. We also perform these control experiments. Basically, in this case we cage the food or controlled object. We use a T-board to cage the food or control object. So in this case, when we present the caged food to the animal actually only gonna receive sensory cue of the food or control object. So basically, the animal only can observe the food or smell the food, but they are not able to approach the food or eat the food. But a very similar data showing the quantification showing here. So those are animal, again, only in the fasted stage. They are selectively increase their calcium transient response in the fasted stage response to the caged food cue. So these experiments actually tell us, so this transient calcium increase actually corresponding, at least partially corresponding to this sensory cue of the food presented to the fasted animals. So through fiber photometry data actually tell us there's a state-dependent difference. In fasted animal versus well-fed animal, their calcium transient are different in vivo. So next, we are trying to understand what actually caused this state-dependent difference. So the first experiment, we are thinking maybe, okay, it could be basically the neuron will be very straightforward. Basically, the neuron firing will change. The intrinsic neuron excitability it could change. To address that, we actually inject a retro AAV virus in the hindbrain, and we selectively labeled those descending projection neurons in the PVH. And through SLAS physiology, we recorded those like EVLP positive GLP-1 receptor neurons actually through many different paradigms. What I'm showing here basically is just inject a current depress the cell. But all of this intrinsic memory property characterization, they all showed negative. So they are basically we're able to conclude that in different states, in well-fed animal versus fasted animal, there are no intrinsic memory property change in this, at least in this descending projection PVH neurons. So this experiment actually narrowed down our scope to where it turns to focus on SNAP transmissions. So by looking at the SNAP transmissions, again, so we injected virus in PBH, and then we recorded this photoactivate response in the hindbrain DVC region. So we simply compare in well-fed and overnight fasted animal. So interestingly, thing is we observed that there actually is enhancements when compare well-fed condition versus fasted condition. So the optogenetic induced the post-synaptic current actually are higher in fasted state versus actually are higher in fed state versus fasted state. So in both, this phenomenon actually showed both AMPA receptor median EPSC as well as NMDA receptor median EPSC, and their ratio actually no change. So put all this data together, we think there's for us, there's a pre-synaptic effect again, actually pre-synaptic synaptic plasticity happening differently in well-fed state versus fasted states. So the next question we are trying to understand, because the GLP-1 actually regulating this synaptic transmission, I already showed in the earlier data. So the hypothesis will be whether this endogenous GLP-1 actually involving this state-dependent synaptic plasticity. So again, to address that, we did a synaptic electrophysiology recording again. So very similar previous slides showed, but in this case, we are compare the example 4 response compare with a well-fed animal versus a fasted animal. Although we already know in well-fed condition versus a fasted condition, the absolute value of a synaptic response is different. But when we normalize the example 4 enhancement data, we're able to quantify and observe there's a selectively facilitation of increase response, increase in fasted states. This descending circuit actually have a better performance, a better effect in responding of example 4 activated GLP-1 receptor neurons. So this actually we think that this in the fasted stage, this descending circuit are actually more sensitive to the GLP-1 receptor activation or endogenous GLP-1 release. So this is a piece of data actually very interesting for us because, compare, I'm showing here this older literature, actually it's a human study in humans as well as in mice. After a large meal, basically when compare the human when in hunger state, when they had a very large meal, they feel full. So there are a dramatic increase in a lot of hormones, including insulin, as well as GLP-1 following a large meal event. So this all the human physiology data actually support our idea. This endogenous GLP-1 level may involve this state-dependent synaptic plasticity phenomenon, what we observed in this PVH to DVC circuit. So next, we're wondering how about in sort of disease states? When the animal become sort of obesity, what happened in these descending circuits? Is again similarly approach. We actually feed the animal for over 12 months. We use a high-fi dye to make versus a control dye. We make the animal to form sort of obese phenotype, and we did electrophysiology experiments again. So in this case, when we recorded this descending response, actually we observed this opposite phenomenon. When animal become obese, high-fi dye, we observed that there's a significant reduction in AMPA receptor median EPSC, as well as NMDA receptor median EPSC. There's no change in AMPA NMDA ratios. That's again indicated this for us is a synaptic effect. And there's for us, there's high-fi dye actually there's a blunting effect actually largely reduce the synaptic transmission through hypothalamus PVH descending to the hindbrain. So the next questions we also test whether in this obese animal whether this state-dependent synaptic plasticity still exists in this obese animal. So by IFIS data, what we collected, we actually observed there's no change, no difference when animal become obese compared with well-fed condition versus fasted condition. Although the absolute value of this EPSC actually relatively much smaller in compared with regular trial-fed animals. So basically, when the animal being fed with high-fi food, they become obese. So they lose the state-dependent synaptic plasticity. And this phenomenon, whether through endogenous GLP-1, whether their mechanism is through endogenous GLP-1 signaling, there's actually, we think there's two major possibilities. We form the two hypotheses. The hypothesis one will be, actually, it will be very similar as leptin receptor work. So this animal may form sort of GLP-1 resistant in this obese animal. So this receptor actually not sensitive to endogenous GLP-1 anymore. So that actually can explain why the animal actually lose this sensitivity or state-dependent change. Also, there's also the second possibility. It could be the endogenous level of GLP-1 actually significantly reduced in this high-fat dye-fed obesity animal, also can result in these phenomenon. So to address that, which hypothesis may be right, so we actually, again, we conduct the example experiment in high-fat dye-fed animals. So through our data, by applying this example agonist to these high-fat dye-fed animals, so although, again, this amplitude is relatively small, but when we apply the example, we still are able to facilitate, increase the AMPA median EPSC. So this piece of data really tell us we actually, is supporting our second hypothesis. Those GLP-1 receptor actually still in this obese animal still responding to the GLP-1 receptor activations. Although it is highly possible endogenous level of GLP-1 their dynamic change are blunted, actually resulting this state-dependent change are gone in these obese animals. So to further understand, all these experiments conducted in slice physiology to further trying to understand also of the blunt effect of a high-fat diet or obese state, we also conduct behavior task. In this case, it's again very similarly, we inject the channelopsin in PVH and we implant fiber in the hindbrain. So, but in this case, we actually conduct relatively more controllable behavioral task, so-called head-fixed sucrose leaking task. So in this case, I'm showing the data here. So through this open loop of the genetic activations in 5 minutes off and 5 minutes on with 20 Hertz light activation, and another 5 minutes off behavior test, we're able observe actually compare with a control group. In the channelopsin group, in the normal trial condition, again, activation of this descending circle is largely reduce their sucrose leaking behavior. But when we actually apply the 12 weeks high-fat diet treatment to the same animal after 12 weeks, we did the behavior test again. So after 12 weeks, when the animal formed obesity, and we actually, when we shine in the light, activated this descending circle, we sort of lose the feeding suppression effect in same group of animal. This also, this data also indicated this high-fat diet also at the behavioral level also has a blunting effect, actually suppress the feeding suppression effect for the optogenetic activations. So next, we're wondering how about the analog of the GLP-1? So for example, mimicking these people or obese patients are taking this analog the GLP-1 analog. So we actually did this IP injection liraglutide experiments. So again, when we feed those animal over 12 weeks high-fat diet, when the animal become obese, we did a very simple treatment. Basically, inject this liraglutide also one type of GLP-1 receptor agonist to IP inject to this animal. After 2 hours, we did an electrophysiology recording again. So through this high-fat diet fed liraglutide experiments and the quantification showing here. So when we inject liraglutide to this obese animal, it sort of can partially recover the synaptic stress in this high-fat diet obese animal. So the AMPA receptor EPSC and NMDA receptor median EPSC are largely significantly increased. And the ratio, again, there's no change. For us, it's a presynaptic effect. So this piece of data actually really aligned with a Nature paper actually published last year. So they actually did liraglutide IP injection experiments. It's very similar than what we did in our paper. So they actually injected liraglutide and did a whole brain C-force mapping. So through this morphology staining data, also the C-force actually represent is an immediately early gene, represent actually activation of neuron activity. So you can see the heat map showing here, across different brain region, so including the hindbrain NTS region and the DMV and the iris postrema, these 3 brain nuclei actually really light up and stand out through their quantification. So through this Nature paper's data actually also supporting our data in this unique brain complex region. Actually, they have this access through this GLP-1 analog can through peripheral injection interact with these hindbrain regions and increase their neuronal activity in the hindbrain. So the next question for us, so whether we saw all these gain of function phenomena in this pharmacological treatments, as well as the optogenetic or chemogenetic activations. So whether actually we want to try to understand whether this descending circuit actually the functioning is important or required for the food intake behavior, as well as the energy homeostasis control. So we decided we want to do a loss of function experiments. Basically, we want to block this synaptic transmission in these descending circuits. So to selectively genetically ablation or block the synaptic transmission in this descending circuit, again, we use our strategy by injecting the ritual on FLIPI in hindbrain, and we inject a toxin virus, so-called tetatoxin, in the PVH hypothalamus region. And this tetatoxin, actually, their function selectively abolish the synaptic vesicle, the snail protein complex function, and specifically abolish the synaptic transmission. So those tetatoxin expressed neuron, they still survive or live in vivo, but they are not able to release neurotransmitters. So through this approach, we actually chronically we observe that in compare tetatoxin expressed group versus control group, we observed after chronically many weeks of recording. So we observed that this tetatoxin group animal actually gain more weight and also increased their daily food intake levels. And also importantly, in this tetatoxin expressed animal, we also observed chronically in the end stage, they also increased their blood glucose level, as well as they increased their liver weight and part of white adipose tissue weight. So all those pieces of data chronically are supporting when we inactivation or ablation of this descending circuit can disrupt or impair the energy homeostasis. You cause the animal form sort of gain weight phenotype. So the next questions we're trying to understand how about the GLP-1 receptor neuron, its function itself, whether this GLP-1 receptor actually is required for this descending circuit. We decide to do another loss of function ablation experiments. So in this case, we're using GLP-1 receptor flux animal, and we selectively inject the ritual FLIPI virus hindbrain, and then we injected the FLIPI on either Cre virus or control EVLP virus in the PVH. And through our quantification showing here, actually, when we selectively knock out this GLP-1 receptor in this descending circuit, we observe that this animal actually also gain more weight and increase their daily food intake levels. So this piece of data also aligned with tetatoxin data. So indicated this GLP-1 receptor function itself actually is important and required for the body weight control as well as the daily food intake level controls. So by putting all this data together, we are able to summarize our study. Actually, we showed this whole story. Actually, we showed is a state-dependent snap plasticity in this descending circle from PVH project to the DVC regions. So you can see, based on our electrophysiology data, we showed this descending circuit. Actually, this is a glutamatergic descending circle. Those GLP-1 receptor neuron project to the DVC region, and the synaptic release actually can be regulated by the GLP-1 signaling. So the GLP-1 signaling can enhance their presynaptic vesicle release probability and the quantum release rates, and actually involve the state-dependent movement, state-dependent food intake control, and energy homeostasis. So we are also able to show this state-dependent synaptic plasticity also regulated by the energy homeostasis itself, also mediated by the endogenous potential GLP-1 signaling. So by this loss of function experiments, including tetatoxin and the knockout experiments, we're able to show loss of function of GLP-1 signaling and the ablation of a GLP-1 receptor neuron synaptic transmission in these circuits. Actually, it can cause the body weight gain and important to energy homeostasis. So with all this data, in the end, I want to thank my previous lab, my supervisor, mentor, Zhiping Pan at Rutgers, as well as all the Pan Lab members, their support in this study. So I also want to thank all our collaborators, including Harvey Greer from UPenn, and also Mark Rossi from Rutgers. Their conceptual input actually is extremely important in guide these studies. as well as I want to appreciate all the funding resources in supporting my pre-doctoral and postdoctoral stage through across all the different stages of this GLP-1 work. So with that, I would like take any questions if you have.